Cellestia basically is a spin-off from the ETH Lausanne, and within ETH there is the Swiss Institute for Experimental Cancer Research, which is one of the high-profile cancer research institutes worldwide, and in this institution, Professor Radke and Rachel Hall have spent considerable time over the last 9 years working on this project in the academic environment before they decided in 2014 to set up a company to commercialize or to develop and finally commercialize the drug coming out of this research efforts. In the end of last year, myself and some colleagues, some of them are here, Dirk is here, joined the company to move it forward and complementing the founder competencies with development expertise from industry. We moved the company to Basel and achieved quite a few really important milestones like listed here, and I'll take you through the steps in the course of the presentation. So if you look at this founder team, this is part of the founder team, not everybody, but the core team which is currently driving the development work forward. This already represents development and research expertise required as co-founders, which also means we don't have to hire consultants for these activities. Of course, each of us has a long-standing track record of success in industry and or research over many years. This is further complemented by a high-profile network of business advisors who support us with fundraising, with finance, with IP, with legal. So again, this is all well covered in the infrastructure, and some of them are actually co-founders. In terms of science, we have also broadened our access to scientific advice on the scientific advisor board, as well as, most importantly, now for moving the project to clinic next year, the clinical advisor board. If you are familiar with oncology, you probably know all these names. This is the top league of European oncology, who are actively working with us to prepare the phase 1/2A program, and in particular with the colleagues at Clinic Vall d'Hebron in Barcelona, we are preparing not only the trial but already now running the biomarker development, which allows us to select the right patients for our trials. Just a bit of housekeeping, as I call it, IP. Of course, we have IP. We are actually proud to have already US and Singapore patent granted. Another set of companies is following soon: New Zealand, Australia, and Israel. This is coming in the next couple of weeks. It's not yet published, so it's not yet on green. In addition, during the process of scale-up manufacture, we had some interesting challenges in our way, which we were able to overcome, which allows us to file a manufacturing patent next year, which is going to expand the patent lifetime by another 5 years. So we're quite comfortable in the patent situation right now. And of course, targeting pediatric and orphan indications, we also expect some help in respect of patent and data protection extension. On the pathway, Notch pathway is known for many years. It is, to keep it simple, a communication between cells that are directly touching each other, and by this contact, an activating ligand is sending a activating signal to the signal receiving cell, which is, for the less scientific people, depicted in this cartoonic thing here. It's like basically a satellite sending a signal to a satellite dish. Then the signal is converted and you see a movie. And the key thing for us is our compound can switch off the cancer movie. It is basically working in the cell nucleus here. It's not acting at the periphery like some of the other competitors are working on. It's actually centrally targeting the pathway and completely switching it off at this level, which means we don't care by which of the activating factors the pathway is triggered, because the complexity is that there's 5 different satellites, 4 different satellite dishes, and this makes the whole thing really complicated. And if you look at the next slide here, this is a different way to describe it on a flow chart, and you see here the activating ligands, the receptors, the intracellular domain, which is released by gamma secretase, and then sends a signal to play the movie, and the constitutive activation, which is basically the pathway activation without any of the periphery engagement. And you see, our competitors are dealing with one of the activating ligands, DLL3 or 4, or a not comprehensive coverage of the receptors, or the gamma secretase, which is really important to launch the signal cascade through this pathway. But of course, gamma secretase has about 70 or 80 other targets and functions in the cell, and if you block that, it is basically not really helpful, because we have seen in a couple of years that one compound after the other is falling off companies' pipelines, because the gamma secretase has on-target but off-notch side effects. In contrast to that, we have basically the most comprehensive targeted therapy on offer, which is controlling the pathway at the cell nucleus. In terms of efficacy, a few examples here. It works in breast cancer, and this is triple-negative breast cancer from a patient which cannot be treated by any of our competitor compounds, because this is one of these cases where the disease is driven by constitutive activation, and there's no targeted therapy available. These patients are typically treated with various chemos. They don't really work, and patients have an extremely short survival prognosis. And here we can show that tumor sizes on treatment, as well as complete suppression of tumor growth on treatment with our drug, at basically no side effects on the test animals. Most excited we are about our collaboration with the Children's Hospital in Zurich, with Professor Jean-Pierre Bouquin. He made it possible for us to access patient blood samples, and this patient samples came from children who died at the age of 3 to 5 on TLL leukemia, and basically nothing worked. The standard therapy doesn't work, chemo doesn't work. These children have been through a number of experimental therapies and finally died, and we were able to, through this collaboration, to get blood samples from these patients, which we incubated over time and at different concentrations, which resulted in this quite impressive kill curve, which basically tells you that our drug, single agent, kills the leukemic cells in blood of these patients, but it only affects the leukemic cells. It doesn't do anything on the other blood cells. We also, in our toxicology studies, don't see any hematological toxicities. So it's really selective just on the leukemic cells. And if you do the typical experiments, transplant these cells into mice, you see on treatment an overall survival benefit and some dose response tested here. Most important for us really is that if you look at these patients, they receive chemotherapy. The chemotherapy doesn't work, but if you take the blood and test the compounds, of course therapy, the chemo at some dose will work, but this is going to kill the patient. Now, what we have seen here is that in combination with our drug, we had an amazing synergy, augmenting the potency of chemotherapy by a factor 700 to 1,000 for two different standard chemotherapy agents. If you think that this therapy is curative in about 70% of patients, and those patients who are refractory or relapse are those patients where this notch pathway plays a really important role, we are hopeful that we could actually develop a curative therapy in combination with chemo for these children and other leukemia. So this is what's really exciting for us and really motivates us to move forward and bring this compound into clinic as soon as possible, which we are working on. Basically, during the course of this year, we have completed manufacture scale-up. I mentioned this is yielding another patent. Formulation work is well on track to be finished in time during this year, and we are about to finish the toxicology and safety pharmacology studies. We know already the data, but we have to wait for the reports. We're going to write up the IND CTA application and then file for clinical trial application towards the end of the year or early next year, depending on how quick the reports are coming in. So we will start clinical development during 2017 during spring. This is showing the relevance. People with Notch die fast, and there's not much that's going to stop that until new therapies come out, and we are working on that. Clinical trial design, standard phase 1 escalation, launching into a multi-arm phase 2 extension arm to stratify for different indications. And this is the development plan. We're currently preparing for a significant fundraising for spring next year to basically finance the clinical development program. And we have been able to tick off all the milestones so far. We still have a small seed C round open, so for people who can rapidly decide, very welcome. Basically, the company is financed completely privately right now, private investors. 50% of the investor base are pharma executives, people who know what we do, and actually many of our consultants invested. So we have a high qualified investor base already engaged, and with that we're looking for private investors as well as institutional family offices, venture capital, etc. Thank you for your attention.